Greetings. Welcome to the NERIC Lightning Talk sessions. This is track 2, and it's going to include the biology sessions, the cancer sessions, and finally the microbial sessions. And so we're really excited. We have a whole bunch of speakers who are going to do our lightning talk format with us here. Before we get started, I just want to describe a little bit about what's going to happen. There is a countdown timer that the speakers will see. So for all you speakers, that will count down when you start your talk from 3 minutes. Please try to respect the time. If anyone has questions, please enter them in the Ask Your Questions box on the next to the live stream. That will be monitored, and if there is time, we will ask speakers questions. And so please, in those questions, indicate who the question is for if you enter a question, and I'll be monitoring that. One other announcement I wanted to make, I know there was one session in yesterday's session, the NAPI talk. The slides for that have been posted on the meeting portal. With that, I think I'd like to go ahead and begin introducing our speakers for our lightning talks. So I will do my best with pronunciation of names, and I'm just going to introduce your name and your institution, and then we'll get started. All right, are you guys ready? I'm ready. All right, our first talk in the biology session is going to be Yuki Shindo from Dartmouth College. Take it away, Yuki.
Thank you. Hi, everyone. My name is Yuki Shindo, and I'm a postdoc in the INBRE lab at Dartmouth. So histones are essential components of chromatin, and they are pretty much well conserved from yeast to human and regulate many aspects of chromatin function. And production of histones is tightly coupled to DNA replication, and any excess of histones gets rapidly degraded in normal cells. However, it is known that excess histones accumulate in some cell types, including cancer cells, resulting in genomic instability and DNA damage. And interestingly, as shown in the cartoon on the right, we have previously shown that excess histones accumulate also in the early embryos and play a role in regulating cell size. However, it is unclear how excess histones feed into cell size control. But because histones are a component of chromatin, a compelling hypothesis has been that their primary effect on cell size would be through changes in chromatin state and/or transcriptional regulation. However, this idea has not been well investigated. Next slide, please. So we decided to ask a very simple question, which is whether or not chromatin incorporation is really essential for histone regulation of cell size. So we made constitutively non-DNA bound histones that cannot be incorporated into chromatin. We then expressed these truncated histones in the early embryo and asked if there is any change in cell size phenotype. And in this study, we measured cell cycle durations as a readout because the longer the cell cycle becomes, the larger the cell size will end up with. And what we found is that, as shown in the right, cell cycle durations were significantly shorter when we expressed H3 tail. And these data indicate that excess H3 tail that cannot bind the chromatin is actually sufficient to affect cell size independent of chromatin. But how is it H3 tail that cannot bind the chromatin can affect the cell cycle? Next slide, please. Okay, so very long story to short. What we found is that histone H3 tail can act as a competitive inhibitor of CHEK1 that controls cell cycle progression. As shown in the gel in the middle, we found that CHEK1 activity is reduced by the presence of H3 tail in a concentration-dependent manner. And this CHEK1 inhibition by H3 is pretty interesting, given the fact that CHEK1 inhibition has shown a therapeutic potential for cancer. And so now we are trying to understand the role of H3 and CHEK1 in tissues outside of the embryo. With that, I'd like to thank the members of the INBRE lab at Dartmouth, as well as funding, including the COBRI grant. Thank you for listening.
Right. Thank you so much, Yuki. That was perfect. You were right on time. So you've set the bar and the example. So I appreciate that. Our next speaker for the biology session is going to be Brett Graver from University of Delaware. Take it away, Brett.
Hello, my name is Brett Graver. I am a graduate student at the University of Delaware in the Southern Lab. Today I'll be talking about bacterial cytochrome C biogenesis, highlighting the biochemical properties of conserved features of the hollow cytochrome C synthase, CCSPA. Cytochrome C biogenesis is the covalent attachment of heme to apocytochrome C achieved via one of three systems, and cytochrome C are unique compared to other cytochromes due to this covalent attachment. System 2 is comprised of CCSPA, where it's proposed to transport heme across the membrane into the periplasm, where it orients it for apocytochrome C attachment. CCSPA itself has a low sequence identity across species, however, maintains key conserved features, including 2 transmembrane histidines, 2 periplasmic histidines, and a WWD domain. We seek to understand if key functions of CCSPA are conserved across bacterial species to determine if CCSPA is a novel target for antimicrobials. Next slide. We hypothesize that CCSPA has conserved function across bacterial species, and using a recombinant expression method in E. coli with its native System 1 CCM proteins knocked out, we're able to investigate different biogenesis systems. A cysteine mutant library across the conserved WWD domain of CCSPA has been achieved, and a cysteine heme cross-linking approach has been taken. When these mutant cysteines come in close contact with heme, a cross-linking event occurs, covalently trapping the heme in the WWD domain. Previous work in Helicobacter hepaticus has identified 6 residues that were able to successfully cysteine heme cross-link and have been identified as heme-handling residues in the WWD domain. Preliminary work in Campylobacter jejuni, a serious threat for drug resistance according to CDC, indicates that preliminary results indicate that these homologous residues, 6 homologous residues, also have the ability to cross-link and are heme handling as well. Future work will include expanding the research into the full WWD domain of the Campylobacter jejuni to further confirm these conserved function across bacterial species. Next slide. This brings us to the 3 biogenesis systems, System 1, System 2, and System 3, and its ability to be a novel antibiotic target. System 1 and System 2 are found in prokaryotes and have similarities. However, there are stark differences between System 1 and System 2 and the eukaryotic System 3. System 3 lacks the WWD domain and requires additional recognition features of apocytochrome C for heme attack. This brings the opportunity about for the development of antimicrobials that can successfully target System 1 or System 2 without the potential of interference of the eukaryotic System 3 biogenesis system. And with that, I would like to thank the funding, the COGRI grant, the startup given to MCS and the Sutherland Lab. Thank you.
Thank you, Brett. That was great. All right, moving along to the 3rd speaker. Our next speaker is going to be Justin Blaze from the University of Rhode Island.
Hi, my name is Justin Blaze. I'm a PhD candidate in the Howlett Lab, and today I'm going to be talking about a novel link between Fanconi anemia and the retinoic acid signaling pathway. Fanconi anemia is a rare genetic disease characterized by birth defects and increased risk for bone marrow failure and cancer. There are 23 FA genes, and the proteins encoded by these genes play a key role in DNA repair and the maintenance of genome stability. Our lab particularly is interested in the FANCD2 protein, which plays a central role in the pathway, although the function is poorly understood. To learn about the function of FANCD2, we recently performed RNA-seq analysis of FAD2 patient cells, mutant for FANCD2, and the same cells complemented with wild-type FANCD2. Euclidean distance, as you can see on your left, and principal component analysis, as you can see on your right, replicates of our replicates demonstrates the expected clustering of samples. PCA analysis shows the most variance, approximately 85%, occurs between FANCD2 genotypes. Next slide. And now you can see a volcano plot that depicts a total of 1,458 differentially expressed genes between the lines. Among many interesting findings, gene ontology analysis revealed differential expression of many genes of the retinoic acid signaling pathway. As retinoic acid signaling plays a major role in development, this pathway was of particular interest to us. 2 genes in particular that were significantly differentially expressed are ALDH1A1 and RDH10. RDH10 converts retinol to retinaldehyde, while ALDH1A1 converts retinaldehyde to retinoic acid. It's important to note that retinaldehyde is a potent genotoxin and can cause DNA damage and genome instability. Over the summer, we have validated these findings using immunoblotting. I want to draw your attention to the 2 lanes marked in red and blue that show our FA test lines, only one of which expresses FANCD2, as you can see in the top panel. In the FAD2 patient cells mutant for FANCD2, we can see a greatly elevated levels of ALDH1A1, and in contrast, no differences in the levels of ALDH2, another aldehyde dehydrogenase we observed between the lines. Next slide. Similarly, we confirmed increased expression of the RDH10 alcohol dehydrogenase in FAD2 patient cells mutant for FANCD2. In conclusion, we have successfully validated altered expression of the retinoic acid signaling pathway in FAD2 patient cells, and these results are highly significant given the pervasive developmental defects that frequently arise in FA patients. And our future directions will attempt to determine the functional significance of altered retinoic acid signaling. And with that, I would just like to thank you for your attention and thank all members of the Howlett Lab for helping me with this and acknowledge our funding sources.
All right. Thank you very much, Justin. So the next speaker is going to be Hui-Jun Park from Brown University.
Thank you, moderator. Good afternoon, everyone. My name is Hui-Jun Park, and I'm a master's student in Brown University. And today I'm going to highlight my research project, Disruption of Putative ABC Transporter B family member in Plasmodium falciparum alters response to anti-malaria drugs. So malaria is the most important parasitic disease of human because of the low efficiency of vaccines and emerge of the drug resistant. New anti-malaria agents discovery and development are urgently needed. The ATP binding capsid ABC transporter carries essential nutrients and is considered the animal cell multi-drug resistant genes. The ABC carriers is also linked to the drug resistance, making it target of effective drug design for malaria. So in the malaria parasite Plasmodium falciparum, ABC transporter B family member has been shown to be involved in the drug response. As below the chart, you can see the information about my genes. Next slide, please. So in this study, I established a double crossover knockout gene, and the human dehydrofolate reductase is the gene encoding. So I construct the plasmid and then put it in the wild type parasite, start to integrate, and it's open reading to the parasite gene, and they cause the recombination. So gene is kicked out, and the few of the base pair are still in there, and it's completely disrupted and human dehydrofolate reductase plasmid construct. And on the right side of the figure, I check the parasite response to anti-malaria drugs, and in particular, the mutation and amplification has been shown to associate with parasite response to artemisinin. Next slide, please. So in the process of immunizing pattern optimized to fragment of mouse model DNA vaccines, and in the right side, the peptide vaccine, I screened the entire genome of Plasmodium falciparum for tandem repeat, hydrophilic domain, and finally, 2 vaccines are ongoing immunized. So we will harvest both of the serum next month and check the antigenicity and the localization to consider vaccine candidate in the future, and evaluate the function of the ABCB6 malaria parasite life cycles. So by doing so, this is my study may provide the basis of the delivery for the new target of the anti-malaria drugs or vaccine and its development of the therapeutic strategy for overcoming artemisinin-resistant Plasmodium falciparum malaria. Thank you for the attention, and that's all.
Thank you very much, Hui-Jun. Our next speaker is going to be Jacob Andrzejczyk from University of New Hampshire.
Thank you. My name is Jacob Andrzejczyk, and in this talk, I'll be presenting preliminary studies on the inhibition of the SARS-CoV-2 main protease by covalent modifiers. So the main protease of COVID-19 is responsible for the cleavage of polyproteins, and inhibition of its cleavage mechanism is likely to prevent the spread of the virus in the host. So we investigated TDZD derivatives, which are known covalent modifiers of cysteine residues in RGS proteins. And in the bottom right, we present the generic structure of a TDZD molecule, where R1 and R2 are functional groups that can be either aromatic or aliphatic carbon chains. In panel A, we present important structural features of a protomer of the enzyme, including the 3 domains in the catalytic dyad of histidine and cysteine. In panel B, we present a zoomed-in representation of the active site with several key residues, as well as the catalytic dyad of histidine and cysteine shown once again. Next slide, please. To probe the effect of these TDZD derivatives on the enzyme, we performed structure-based docking and long-scale all-atom molecular dynamic simulations. Notably, we calculated the change in root mean square fluctuations of all residues in the protein, and we reported that the majority of active site residues were stabilized in the presence of the ligand, while those residues located on the outside of the active site were actually destabilized. Interestingly, we noticed increased fluctuations in domain 3 of the protein, which is far removed from the active site, suggesting a potential allosteric effect of the ligand. Additionally, in the apo form of the enzyme, we observed a flipping mechanism of the catalytic histidine residue, indicated by panel A, with 2 prevalent states, B and D. In the liganded form of the enzyme, we noticed that these states became less prevalent, indicated by F and H, and a third state, G, became the most prevalent state. It is known that the cleavage mechanism of the main protease is initiated by a proton transfer from the catalytic cysteine residue to the catalytic histidine residue, so any change in the orientation of the histidine residue is likely to inhibit this mechanism. Next slide. Further, we are interested in studying the effect of the ligand on global protein dynamics, so we performed a community analysis on the apo and liganded form. The community analysis offers insight to correlations between residues on a local and global scale. Most notably, we noticed that community 9, which contains the catalytic cysteine residue, increased in size, so had increased correlations to neighboring residues, and saw increased correlations to domain 3, which is far removed and contains communities 3, 8, 6, and 10, offering insight into that potential allosteric effect observed in our root mean squared fluctuation calculations. Furthermore, we performed protein expression assays to derive the inhibitory concentration of our ligand, and we report inhibitory concentrations on the micromolar scale, comparable to known inhibitors tideglusib and carmifer. Thank you for listening to my talk.
Okay, thank you very much, Jacob. All right, then the next speaker in the Lightning Talk biology session is Min-Jae Kim from Dartmouth College.
Hi, everyone. So my goal is to investigate the binding interface between the paired plasmic domains of TOX-RS complex from Vibrio species. And currently, importantly, the cholera has about 4 million cases and about 143,000 deaths worldwide every year. And in a case like Vibrio cholera, TOX-R is required for human pathogenesis. And TOX-R is a transmembrane transcription factor, and TOX-S is an integral membrane binding partner of TOX-R. And TOX-R requires TOX-S in order to be fully active for virulence gene expression. And it's also necessary to protect TOX-R from premature degradation. And the complex of TOX-RS is important to understand the virulence of these bacteria. And on the right is a model of showing how TOX-R and how TOX-S interact together. And these 2 proteins interact as their paired plasmic domains. And so far, we saw the structure of TOX-R paired plasmic domain, and I'm interested in the structure of TOX-S paired plasmic domain, which is required in order to understand how they interact. Next slide, please. So we were able to successfully purify the TOX-S paired plasmic domain using the sumo tag, and this protein with the sumo tag together shows bands at around 35 kilodalton. And when we add the sumo protease, the cleavage occurs immediately. And as a result, we purified the protein at about 17 kilodaltons. And interestingly, we noticed some differences when we ran the gel with the crystals and the input mixtures of the TOX-SP. So we examined them through mass spec, and interestingly, that we found that this protein experiences a secondary cleavage, and the possible cleavage site is indicated with arrows, and it appears that TOX-SP that's cleaved from an intine tag is the one that has the full length. And to solve this secondary cleavage problem, we tested with our protein with different types of protease inhibitors, and the N-ethymelamide, noted as NEN, is the one that seems to inhibit the secondary cleavage. And next slide, please. And the structure that you're seeing here is the potential TOX-RS complex structures that's aligned to BTR-AC complex, which we believe are homologous with each other. And the structure in pink is TOX-R periplasmic domain that we saw previously in the lab. And the structure in blue is TOX-S periplasmic domain that's predicted from AlphaFold. And we crystallized TOX-SP from Vibrio parahemolyticus, and we collected X-ray diffraction data at about 3.2 resolution. And we hope to solve the phase problem using the selenium methionine labeling or co-crystalline complex. And thank you for listening to my talk.
Thanks, Minjay. The next speaker in Lightning Talks is Nathan Angel from Bryant University.
Hello, everyone. My name is Nathan Angel, and I will be presenting on connecting the dots, deep learning and its application to identifying chromatin regulatory loops. Our research began this summer with working on zebrafish embryos to study the 3D genome structure. And this 3D genome structure consists of chromatin loops, which is important in gene expression and normal cell function. And we know that any changes to this structure can lead to diseases such as cancer. As of now, we know very little about these chromatin loops and how they are controlled in the cell. So we used a process called 4C, located in the bottom left, to try to look at how chromatin is formed and to thoroughly study it. This process separates these loops in order to discover how chromatin is ordered and regulated through mapping and sequencing. And we believe that as we study the different time points in the zebrafish embryo development, we can potentially reach our goal of better understanding the gene expression and how chromatin is organized within the genome. Next slide. So with this in mind, the 4C process generates a vast amount of data that is generally not organized. So we decided to create a data pipeline to resolve this issue. And this data pipeline organizes it and gets rid of any unwanted data that's we do not necessarily need. Next slide, please. In addition to the data pipeline, we are going to program the pipeline to output these kinds of images shown all the way to the left to see which genes are close to each other visually. Next, we will input these images into our deep learning model, our convolutional neural network, or CNN for short, that we believe will have the potential to explore different connections that we could have not seen in the conventional 4C analysis. CNNs are a special type of deep learning neural network that specializes in computer vision or letting a computer understand any kind of image. CNN has the capability because of its hidden layers, shown in the middle picture with the blue dots. The fact that it has these hidden layers allow it to learn as many input images the data set has to offer, of course, without compromising the accuracy of making predictions and learning what the input images are. This concept of adding any amount of input images without compromising accuracy is very unique to other similar models out there like random forest decision trees. That is why CNN was chosen, because removing data is something that we do not want to do. Anyway, thank you.
All right. Thank you very much, Nathan. Our next speaker in Lightning Talks is Laura Drapanos from Colby College.
Hello. So the trajectory of our health as we age is largely driven by what we are exposed to before we are even born. Chronic stress during development can cause individuals to have high inflammation, and this is the root of many degenerative diseases. Since the Coffin Lab has identified that this cause and effect does not occur in zebrafish lacking the transcription factor KLF9, identifying the effects of this gene can help us become proactive in preventing disease. Based on the knowledge that KLF9 has a deep involvement with the core circadian clock mechanisms, the Coffin Lab at MDIBL captured RNA-seq data in zebrafish with and without the KLF9 gene at 3 hours before, at, and 2.5 hours after the onset of light. My role has been to develop a robust and reproducible computational approach to identifying the effects of KLF9 with respect to time from this data. Next slide, please. So after running principal components analysis to control for experimental noise, I ran differential expression analysis between transcript counts of all genes between the wild type samples and KLF9 knockout samples to see the effect of removing the KLF9 gene on gene expression. I identified some genes that have a time-dependent response to KLF9 loss, such as CRYBB1, which is a visual development gene, which we can see has a positive log-fold change in transcript counts and KLF9 knockouts at 3 hours before lights on, but at no other time, meaning that it's uniquely upregulated by the loss of KLF9 at that time, whereas other genes such as C8b, which is a complement activation gene, is upregulated by the loss of KLF9 at all times. Next slide, please. We can also estimate the biological effect of the loss of KLF9 with gene enrichment analysis. So summarizing the Venn diagrams below, we can identify gene ontology groups or biological processes defined by a set of involved genes that are well represented among genes upregulated or downregulated by the loss of KLF9. The genes that are highlighted in the middle are those that the Kaufman Lab has previously identified as responding to the loss of KLF9, but this is from an experiment that only captured data at one time of day. And those that are highlighted are those from this analysis that were found to have the same effect at all 3 times that data was collected. And since KLF9 has such a time and day-dependent response, this gives us much greater confidence in these prior findings. We also identify groups such as visual perception genes that have a time and day-dependent response to KLF9 loss, and in this case, that's uniquely upregulated by the loss of KLF9 at 3 hours before lights on. So hopefully all this analysis can give us a greater understanding of the mechanisms of KLF9 so we can become proactive in preventing disease. Thanks.
Thank you very much, Laura. Our next speaker is going to be Haley Naiman from Bates College.
Hello, so today I want to talk to you a little bit about stress. So we all experience stress in our everyday lives, and this is not always a bad thing. Stress can be used when we need to wake up in the morning, catch a flight on time, or when we're giving a live presentation. But stress is also a helpful way for our body to regulate itself and respond to external stimuli appropriately. However, we also know that stress can have some detrimental effects on our health as well, especially when we are exposed to a high level of stress during a traumatic event or long-term exposure, which is called chronic stress. So the Kaufman Lab is interested in modeling chronic stress using the zebrafish model organism. To do this, the stress hormone cortisol is used. However, as I will mention later, cortisol is not only used in responding to stress. So much like ourselves, zebrafish express the glucocorticoid receptor ubiquitously, and it is assumed to be the principal mediator for responding to cortisol. I've shown here that cortisol, after it's released by the body into our bloodstream, can enter the cell and bind the glucocorticoid receptor as marked by the GR. From there, it can travel to the nucleus and bind DNA and regulate certain genes, one of which is KLF9. So KLF9 is an important transcription factor, and the Kaufman Lab has indicated that it's very important for responding to glucocorticoid signaling. KLF9 itself can bind DNA and regulate the expression of certain genes. Next slide, please. Without an external stressor, cortisol cycles through a circadian rhythm throughout the day. It also experiences ultradian rhythms every hour. This note became important when analyzing past research by the Kaufman Lab because varying sampling times led to different gene expression results. So to research this, a recent study looked at using wild-type zebrafish and KLF9 knockout zebrafish at 3 time points as shown by the stars: 3 hours before lights on, at lights on, and 2.5 hours after lights on. We analyzed this past experiment using RNA-seq. My research project aims to identify or verify differential gene expression by repeating this study using qPCR. Next slide, please. So to do this, I rose wild-type and KLF9 knockout zebrafish to 5 days post-fertilization, isolated their RNA using Trizol, and then made cDNA to run qPCR. From the qPCR results, we found that many of the genes that we identified were less dynamically expressed in the KLF9 knockout zebrafish than the wild-type zebrafish. The results of this research will illustrate the importance of considering time of day on gene expression and the different ways in which KLF9 mediates this expression. Thank you.
Thank you, Haley. So this is going really fast. I've never done a lightning talk session before, but this is very crisp and everyone's staying on time, and I appreciate that. Our next speaker is Emily Ann Ashford from Keene State College.
Hello, my name is Emily Ann Ashford, as you know, and I'll be talking about the dietary phosphorus mediating arsenic toxicity in Daphnia pulex. So to start out, arsenic is a potential human health concern prevalent across the United States, New Hampshire, and the world, in some cases greater than the EPA threshold of 10 micrograms per liter. We know that arsenic is biotransformed in humans from the trivalent to the more toxic pentavalent via methylation steps. And it's also been seen that associations between arsenic toxicity and malnourishment has been observed in humans. We aim thus to explore the mechanisms underlying the observed interaction effect of nutritional status and arsenic toxicity, specifically identifying physiological, genetic, and metabolic pathways regulating these observations. These findings will hopefully give a mechanistic understanding applicable for risk management and vulnerable populations. Next slide, please. So we define nutritional status as phosphorus supply due to phosphorus. Can you go back a slide, please? Due to phosphorus and arsenic being in the same group on the periodic table, and thus having similar biochemical properties, along with some organisms being able to use them interchangeably. We study this in our model organism Daphnia pulex due to their cost-effective maintenance, fully sequenced genome, and their easily manipulated food source. Studies have tested the effects of phosphorus supply and arsenic separately on Daphnia, but not together, and that's what we study as their interactions. Jumping into the results, we saw how arsenic interacted with phosphorus physiologically, with greater lifespans in arsenic with low phosphorus supply compared to higher phosphorus supply, as well as faster, more variable reproduction in high phosphorus with arsenic treatments versus low phosphorus with arsenic treatments, indicating high phosphorus with arsenic was more stressful. For genetic, we saw a total of 11,185 Trinity transcripts from our de novo assembly, with 3,681 upregulated in the control and 1,799 upregulated in arsenic. These results can indicate an interaction effect mediating organismal fitness and gene expression. Next slide. Looking at the metabolome analysis in the NMDS, there was clear clustering among HP, high phosphorus in green, and LP in yellow, while arsenic in control showed no clustering. Bar plots showed top proteins of 62 differentially expressed proteins in phosphorus conditions on the left, and 15 differentially expressed proteins in arsenic on the right. 6 were common among the two. Half of the differentially expressed genes, as I'm sure you can notice, are uncharacterized due to Daphnia's genome not being fully annotated, and thus we have to do more study to look into the functions. But we have begun to observe metabolic processes mediating the arsenic and phosphorus interaction. For our future directions, we plan to increase our exposure to demonstrate chronic toxicity, which is more prevalent across the world. And then finally, I'd just like to thank NERIC for this opportunity, Keene State College, Sea Grant, SEPA, and New Hampshire INBRE for the funding, as well as my lab. Thank you so much.
Thanks, Emily Ann. Our second to last speaker is going to be Eric Borman from MDI Biological Institute or Laboratory.
Hello. This summer I worked at MDI Biological Lab investigating the relationship between the EIF3 complex and the VASA GLH1 protein in C. elegans. Next slide, please. So globally, about 50 million couples are affected by infertility, and millions of these people can be otherwise perfectly healthy, other than the fact that they're infertile, which kind of brings into question what's causing the infertility. And one answer to this might be germ granules. These germ granules are non-membrane bound organelles that can be found around the nucleus of germ cells and act as a sort of filter, only allowing passage of transcripts needed for germline development. And we don't fully understand germ granules yet, but we do know that a central component of them is the VASA protein, a highly conserved helicase that's essential for both development of gametes and embryos. Now, looking at the VASA equivalent in C. elegans, GLH1 is found to interact with a multitude of proteins, not necessarily part of the germ granule complex. Can I have the next slide, please? Now, one of these proteins was EIF3G. It's the RNA binding subunit of the larger eukaryotic initiation factor 3 complex. And the relationship between EIF3G GLH1 isn't fully understood yet. So this summer, I looked at trying to verify this protein-protein interaction and trying to determine biological significance. So in order to do this, used a strain of C. elegans that had part of the GFP protein attached to GLH1 via a self-degrading linker, allowed for the spatial separation of the two components. Then used CRISPR to tag EIF3G with the remaining portion of the GFP protein, so that whenever the two GFP protein portions came together, there'd be a fluorescent signal. Now, upon successful tagging of these, there was a fluorescent signal throughout the entire germline. This kind of verified some sort of interaction between the two proteins. So our next step was to remove that self-degrading linker between GLH1 and the GFP protein so that the two would now be stuck together, and that, in theory, any fluorescent signal that we now saw would be wherever EIF3G and GLH1 are directly interacting with each other. So we were able to successfully remove that self-degrading linker between GLH1 and GFP. However, we're unable to homozygose it. So this is still a work in progress, but currently our working theory is that GLH1 mediates passage of transcripts out of the nucleus, passes it off to EIF3G, where GLH1 then disassociates, and EIF3G helps initiate translation of the transcript. Thank you.
Thank you very much, Eric. All right, our final speaker of this biology lightning talk session is Linda Miranda Portillo from Bowdoin College.
Good afternoon, everyone. I will be talking about how researching implantation at a molecular level has been difficult within the field of reproductive biology because of the absence of an in vitro model. However, we seek to establish C. elegans as a model for FG repeat-mediated signaling that happens during human embryonic implantation. An overview of the germline of C. elegans shows that after undergoing mitosis and meiosis, we see perinuclear P granules in the adult germline. P granules are specific to the germline and contain the dead box proteins GLH1 through 4. Next slide, please. Past studies have shown that GLH1 contains phenylalanine glycine, or FG repeat domains, which are small hydrophobic segments that break up long stretches of hydrophilic amino acids. FG repeats are found in several nuclear pore complex proteins where they establish the size exclusion barrier between the nucleus and the cytoplasm. In C. elegans, FG repeats can be also found on core germ granule proteins, such as the GLH helicases, where they extend the size exclusion barrier of NPCs into the cytoplasm of germ cells. In humans, a protein called trophinin contains an extremely long FG repeat domain, which is involved in embryo implantation. What we know is that trophinin interacts with another protein called tastin indirectly through an intermediary protein called bistin. Bistin has a homologue within C. elegans called BIN1, whose function is unknown. Our overall goal then was to determine if BIN1 is localized in the germline to begin characterizing it. Next slide, please. To pursue this goal, we tagged BIN1 with a green fluorescent protein using CRISPR, which showed us that BIN1 is mainly in the nucleolus of germ cells and to a lesser extent the cytoplasm. We also conducted an immunostaining to visualize the proximity of BIN1 and the P granules, which showed that there might be some overlap between the two. Our next steps would be to confirm if BIN1 interacts with GLH1. And although we haven't completed this yet, our current results do not indicate that the two do not interact. Since proteins with FG repeats have been investigated for their function, we anticipate that our research on BIN1 from the angle of FG repeats will allow us to understand the role it plays within C. elegans to one day understand a portion of embryonic implantation. Thank you for listening today.
Right, thank you very much. Those were all great. And as promised, they were lightning fast. There haven't been any questions entered in the chat, but I do have a few if you guys might indulge me. So what I'll ask is for the producer to make you live again to answer the question. And my first question is for Nathan Angel from Bryant University. Nathan, you gave a nice talk about mapping chromatin interactions in the genome. And I was wondering if you could comment a little bit more about where do you think that's going? Is there some idea that the three-dimensional location of different parts of the chromosome could be meaningful for chromosome function?
Yes, we do believe that, and that's the reason why we're trying to actually locate where these genes are, because we feel as though if there are any non-similar situations for one cell, that one cell, when it replicates, that will lead to potential cancer, and we all know that cancer is when bad cells replicate like too many times, and we feel as though if we can locate that cell, like bad cells, then that could help us relieve some pain and locate that into one area, because the treatment that we have as of right now, it drops a bomb on the entire human body, and we feel as though if we can just specify it to only one specific area, it's a lot better for it, and that's the reason why we're trying to do that.
All right, thanks a lot, Nathan. I have another question, and this is actually for two of the speakers, Haley Namond and Laura Japanos, because both spoke about KLF9. And so I was wondering if one or both of you had any thoughts about a question that might be a little bit out there, but you both are studying gene expression changes that are responsive to circadian biology, and also there's a link there to cortisol. Is there any evidence that KLF9 expression or function changes at all with organism age, and therefore you end up with changes in how responsive the organism is to something like cortisol signaling? And these questions producers asked me to repeat are for Haley Namond and Laura Drapanos. This is so you guys can be moved on to the stage. Sorry.
No, Haley, you're welcome to talk about it too. We only capture data at 5 days post-fertilization in all three studies that we mentioned. So we've only really looked at KLF, or at least Haley and I have only looked at KLF9 expression at that time. But I could imagine that it could change, but we haven't really focused that much on it. I don't know if Haley, you have any ideas on that?
Yeah, I would just add that we were looking at early life stress and like how cortisol inside a mother could lead to gene expression effects in like a fetus. So that's the level of cortisol we were putting in the zebrafish media, was the same amount that would be exposed to a fetus. So yeah, we were looking at early life stress, but I imagine also that there could be differences in older zebrafish.
All right, thank you guys very much. So I'm going to stop asking questions now. I just wanted to thank all of the speakers. Giving a lightning talk is not an easy thing to do. Certainly stress levels rise during that, and I want to thank you guys for giving great talks and for having organized and stayed within the time limits. I'll remind you guys that there are awards for lightning talks. We have some generous sponsorship from both NAPI, who will be giving one award to each trainee level, as will Driven for entrepreneurship-driven talks. So with that, I'll thank you, and we'll take a short break before the beginning of the second lightning talk session. So thank you, everybody.